Well, good evening, everyone. It's Mrs. Suter. I'm back with our After School with the Science Lady. And today we're going to be talking about biotechnology and DNA. So I want to tie it in with something that you may be familiar with. I know that a lot of us like the movies Jurassic Park. And so what I want to explain to you is what is the science in Jurassic Park, and how does DNA help the scientists to create new dinosaurs or recreate the dinosaurs that were lived a long time ago. And many of these scientists that you would see working actually work in the field of biotechnology. And so we're going to do a lesson about DNA and biotechnology. And the materials that we're going to need for this lesson are some toothpicks, and I use these that have the sharper points on the ends. Okay, and you need about 7 or so of those. I'm using 2 pieces of licorice. These are red vine, but you could use Twizzler or whatever you can find. So I've got 2 pieces of licorice. And I'm using these mini fruit flavored mini marshmallows, because I need something soft that has 4 different colors. And this happens to have 4 different colors in it. But you could use gumdrops, 4 different colors of gumdrops, or you could use gummy bears or something in that line to do it, but I'm just using the mini marshmallows to do mine. So, and you need to have 4 colors. Now, I've already labeled my 4 colors as to what they're going to represent. If you don't have the same 4 colors I do, just change the color to another color, just as long as you're consistent. Okay? So we're going to go ahead and get started. I'm going to start off by showing you a clip from the movie Jurassic Park, and I want you to kind of pay attention to some of the science that they're talking about here at the beginning, okay? And this is just a small clip. All right. Okay. So here we go, and I hope I'm going to turn the sound up so you can hear it. Oh, went the wrong way there. There we go.
Of the entire planet.
So what are you thinking?
We're out of a job. Don't you mean extinct?
Why don't you all sit down? Donald, sit down. Right, here he comes. Well, here I come. Yes.
Hello. Hello.
Say hello. Say hello.
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 Hello, John.
Oh, yes. I've got lines here.
Well, fine, I guess. But how did I get here?
Well, let me show you. First, I'll need a drop of blood. Your blood.
Right. Oh, John, that hurts. Relax, John. It's all part of the miracle of cloning. Hello, John. Hello, John. Hello.
Hello, John. From what?
Hello, John.
Low extractions never recreated an intact DNA strand.
Never thought master sequence gaps.
Good afternoon,
Your
John.
DNA from what source? Where do you get 100-million-year-old dinosaur blood?
Shh.
What? Oh, Mr. DNA, where did you come from?
From your blood. Just one drop of your blood contains billions of strands of DNA, the building blocks of life. A DNA strand, like me, is a blueprint for building a living thing. And sometimes animals that went extinct millions of years ago, like dinosaurs, left their blueprints behind for us to find. We just had to know where to look. 100 million years ago, there were mosquitoes, just like today. And just like today, they fed on the blood of animals, even dinosaurs. Sometimes, after biting a dinosaur, the mosquito would land on the branch of a tree and get stuck in the sap. After a long time, the tree sap would get hard and become fossilized, just like a dinosaur bone, preserving the mosquito inside. This fossilized tree sap, which we call amber, waited for millions of years with the mosquito inside, until Jurassic Park scientists came along. Using sophisticated techniques, they extract the preserved blood from the mosquito and, bingo, dino DNA. A full DNA strand contains 3 billion genetic codes. If we look at screens like these once a second for 8 hours a day, it'd take 2 years to look at the entire DNA strand. It's that long. So, it's full of holes. Now that's where our geneticists take over. Thinking machine supercomputers and gene sequencers break down the strand in minutes. And virtual reality displays show our geneticists the gaps in the DNA sequence. We use the complete DNA of a frog to fill in the holes and complete the code. And now we can make a baby dinosaur.
This score is only temporary. It all has very dramatic music, of course. Well, a march or something hasn't been written yet. And then, of course, the tour moves on.
Okay, so that was a very that was the description of some of the science that was taking place in the movie Jurassic Park. And so today what we're going to do is we're going to take a look at what DNA is. We're going to build a model of DNA, and we're going to talk about where do we get DNA, and how can we use DNA today. Now, we will talk a little bit about cloning, but not a whole lot. But just know that some of the things that happened in that movie could not have happened in real life. But there was also a lot of science that dealt with DNA and biotechnology. And so I thought I would just talk about that today. Okay, so we're going to start off by learning a little bit more about DNA. And we're going to talk about how we use it, where we get it from and how we use it. So when we talk about biotechnology, the word bio, this prefix bio means life. And technology, this part of the word, it means the application of scientific knowledge for a practical purpose. So it is using scientific knowledge to create life. And that's what biotechnology is. So biotechnology is using biological or life processes and systems to make or improve products. Now, yeast is another living organism, and we use it in bread to make the bread rise. And that is another example of biotechnology. We're using something living in a practical reason, a practical purpose for using life. And so that's how we get biotechnology, okay? So what is DNA? Well, this is a picture of DNA that's been highly magnified because DNA is very, very tiny. But DNA in these little strands, these little rungs of the ladder here, carry all the information about how the living thing will look and how it functions. And now, DNA stands for I never can say this word, deoxyribonucleic acid. Okay, it is in every cell in your body. So, and these cells, DNA is found in the chromosomes of the structure of the cell known as a chromosome. Okay, now the DNA is made up of 4 chemicals, right? And they are put together in a very specific way, right? So DNA is very much like how your parents or grandparents would use a cookbook. You put all the ingredients in there, you mix it all up, and you create a new thing. Well, the DNA it stores the recipe or how the organism was built. So even though all of our DNA, all the sequence it comprises 4 chemicals, 99.9 of all of our DNA is identical. So if 99.9% of all of our DNA is identical, how come we're not all the same? How come we're all different? And so we're going to talk about how DNA is put together and how we use DNA. And then you're going to maybe understand why, if there's only 0.1% of DNA that's different about the structure, then how come we are all very different? All right. So I always think about DNA as being the cookbook for how that living thing came to be. All right. So if we take a look at DNA, as I told you, it's what we call the genetic blueprint of life. All right. And here are the 4 chemicals that make up DNA. All right. We have adenine, thymine, guanine, and cytosine. Those are the 4 chemicals that make up the ladder that we call DNA, the genetic blueprint. Okay, it's a very long molecule and it looks like a rope ladder. Okay, so here's your 2 strands, and then in between all the rungs are these 2 chemicals combined together. Now, the DNA that you have here, that we're showing here, is much larger than real DNA. This right here is 200 million times larger than a real DNA would be. Okay, and DNA is twisted in this clockwise twist, and that is called the double helix. And we're going to see why or how the double helix helps. Okay, so now to figure this out, we are going to actually make a model of DNA, okay, a piece of the strand. Now, as I just told you, DNA uses the 4 chemicals to encode or write the recipe, okay? And so these chemicals we're going to abbreviate with the letters A, T, C, and G. And that's adenine, guanine, thymine, and cytosine. Now, these 4 chemicals are joined together in a very specific way, all right? So if you look over here, you can see adenine is in the green, guanine is the blue, thymine is the purple, and cytosine is our pink, okay? Now they're joined to our 2 little rope ladders, or, the 2 rungs over here, all right? Now, after we know that we're going to use colors to represent each of our different chemicals. So we need to have 4 different colors of things, all right? So I'm going to use my marshmallows because they come in 4 different colors. And so what I'm going to do is I'm going to go ahead and start, and I'm going to want you to do this too. I want you to pull out some of your marshmallows or gummy candies or gumdrops, and I want you to begin sorting them in their 4 colors. So I'm going to use the orange. I'm going to separate those out. And then I'm going to put the green over here. Separate those all out. Oh, got a yellow in there. And yellow. And then I'm going to put pink over here. All right. So I've kind of just separated out my colors into their groups. Okay. I'm going to pull myself out a couple more little orange because I don't have a lot of this. All right, so you can see I've separated my marshmallows into their 4 colors. So you need to do that with your whatever kind of candy you're using, gumdrops, gummy bears, marshmallows, whatever, okay? Now, if you only have white marshmallows, you can use the white mini marshmallows and then just color them with a marker. Just remember that afterwards you shouldn't eat them, okay? So go ahead and sort out your candies now. Okay, now I'm going to use this particular structure for mine. My adenine, which is going to be A, I'm going to make that my yellow. So my yellow is going to be my adenine, okay? So I'm going to put a little A down here by my yellow marshmallows. Now my pink marshmallows are going to be the thymine. I'm going to put a T down here. My guanine is going to be my green, so I'm going to put my G up here to stand for guanine. And my cytosine is going to be orange. Okay, so I have these labeled, and I'm going to show you here how I did that. I just put guanine G, cytosine, I put my C because that's my orange, my yellow is my adenine, and my pink is my thymine. Okay, so I just put those little letters up here to remind me of what color I'm using. Okay, so I'm using my paper and a marker to help me out there. Now, the funny thing about DNA is that these 4 chemicals only go together in a certain way. So 2 chemicals are only pair with 2 other chemicals, all right? So they always pair up in this specific way. A, adenine, only pairs with thymine, okay? And C, which is cytosine, only pairs with guanine. So it's always going to be A with T and C with G. They're like best friends and they can't be separated. So I cannot put A and C together, or C and T together, or A and G together, or T and G together. It's always going to be A and T, adenine and thymine, and C and G, cytosine and guanine. So in my model, I can only put my pink with my yellow, and I can only put my green with my orange, because that's the way the DNA is built. All right? So what we're going to do now is we're going to get some toothpicks. And I'm going to use 6 toothpicks here. I'm going to lay my toothpicks out. I got 6 here. And then I'm going to start putting my rungs together in their sequence. All right. So I know that, as we just explained, that cytosine, for me that's orange, will only pair with guanine, green. So I'm going to put my cytosine and my guanine together. Okay. Then I'm going to put this rung down, and I'm going to make 6 more rungs. Okay. And it doesn't really matter what you use. This one I'm going to use my adenine and my thymine because they go together. Okay. Now I've got 4 more rungs to make, and I can, maybe I'm going to make another thymine and adenine. And I'm going to make another cytosine with my guanine. And I'm going to do another one of those too. Another cytosine and guanine. So I got my guanine on there, cytosine on there. Okay. And I think I'll make, oh, I like that one. So I'm going to do that another one of those. Okay. Now you make your 6 rungs however you want to make yours. And I'm going to lay them kind of out here, and I'll show you where I've got them. So here you can see I've got my little rungs, and I have a lot of extra marshmallows, which I'm not going to use right now. So I'm just going to move those over. And I have my rungs, kind of my ladder here. Okay. Now, so these are going to be the rungs of my ladder. So, oh, I need to be able to connect them to something. So that's where I'm going to pull out my licorice, and I'm going to put those on either side. And now I'm going to decide how I want to organize these. Maybe I want to go like this, okay? Or I want to turn that one and maybe turn this one, okay? So now I'm going to put it together. So I'm going to poke this end of my toothpick in to that side of the licorice, and this in here. Okay. And now I'm going to leave about an inch between and poke it in there. Poke it in there. Now I've got 2 of my rungs together, and I'm going to keep going down my licorice, putting the rungs on my ladder there. Okay. I'm going to keep going down. Now I like to push on my edges a little bit. It pushes the toothpicks in a little bit better. All right. All right, so now I have my ladder. That is my DNA. I'm beginning on my DNA. Okay. Now, the only thing that's left is to make the double helix. I'm going to put my fingers here and here, and then I'm going to twist, and now I have a model of DNA. Okay. Notice what happens when I make the double helix. So here, and now when I go that, what happens to it? What happens to the height when I go from straight to the double helix? Okay, and I want you to think about that. All right, so I'm going to lay mine down. We're going to go back to our presentation here for a minute, and we already put together ours. Now, we put our DNA together here. Now on our ladder right here, I have 1, 2, 3, 4, 5, 6 pairs. So in our little model, we have 6 pairs. But in real DNA, the real DNA in your body, it doesn't have 6 pairs. It has about 3 billion pairs. So if you imagine trying to make a model of real DNA, and this is only 6 pairs, and you had to make 3 billion pairs, you're talking about a very, very, very long DNA strand, okay, if we were making this model. And here in a little bit, we will make the, we'll do a little of the math to show you how long that would be if we were making a real model of the whole thing. All right, so now what we're going to do is we're going to take my paper, and I'm going to lay my model down on my paper as rungs, and I'm going to go ahead and start to label my sides, okay? So, got my marker here, and I know that my pink was thymine. So on this side, I got thymine. And I know thymine only ever matches with adenine. So this side is the yellow, the A. Okay. So now here, the green is guanine. The orange is cytosine, because I know G and C go together. Now I've got cytosine here, guanine here. And here I have the yellow, which is adenine. And this is the thymine. And then I've got green, which is my guanine, cytosine, cytosine, guanine. All right, so here in my DNA strand, I can see that on my left-hand side, I've got TGCAGC. And on my right-hand side, I've got ACGTCG. So if we were to all of us compare our letters, I bet you there is no 2 people in this room that would have the exact same sequence. So even though we're using the same 4 chemicals in the DNA, none of us have the exact same sequence as another person. So even if I did this a billion times, I would not have the same sequence. And since I wouldn't have the same sequence, then 2 people can't be exactly alike. And that's the thing. And this is only a little teeny piece of the DNA that's in our body. This is only 6 pairs. Imagine trying to match up exactly 3 billion pairs. That's not possible. And so that's why all of us, even though 99.9% of our DNA is exactly the same, the sequence is always different. And since that is, all of our, all living beings look different. Okay, so now I can take a look at my DNA and I can fill in my sequence, and I even know that T always goes with A, thymine always goes with adenine, and guanine always goes with cytosine. They cannot match up with anyone else. Okay, so now that we've got our DNA built, then like I showed you, we made the double helix, so we twisted it. So I just kind of twisted. Okay, and what happens when I create that twist? All right, so I want you to think about what happens to the length of this DNA when I twist it and make the double helix. So first, when I have my DNA and I measure it, it is 8 centimeters long. Okay, now when I twist it into the double helix and I measure it again, now I have it 6 centimeters long. So when I twist it, what happens to the length of the DNA? It gets shorter. So the twisting allows the DNA to get more strands in there in less amount of space. Okay, so if we know this is just a model of our DNA, this is not the real DNA. The real DNA is way tinier than this. But if I were going to build a model in this way of the full DNA, we're going to do some math to figure that out. How long would our model be if we actually modeled all 3 billion pairs of the DNA? Okay, so I did put some of the math over here, but I'm also going to stop sharing, and then I'm going to bring up the calculator, so we can take a look and do some of this math, because this is not math that's easy to do in our head, okay? So I have my calculator here, so let me go back in and share so you guys can see my calculator. All right, so we know that our model was 8 centimeters, okay? And in 8 centimeters, I could get 6 pairs. So I need 3 billion pairs. So that means I need to take 3 billion and divide it by 6. And that gives me 500 million more pairs that I would have to make. So that would be 500 more million pairs just like this. Now, if each of these strands is 8 inches and I need 500 million more of them, I would have to take my number here, 500 million, and times it by 8 centimeters. Or 8 inches, sorry. That would mean that our strand of DNA would be 4 billion inches long. And if I want to know how many feet that is, I'm going to have to divide it by 12 because there's 12 inches in a foot. So that would mean if I modeled the entire strand of 3 billion pairs of DNA, it would be 333,333,333 feet long. Now, to put that in perspective, that would be like making this so long that it would wrap around the entire world 1.5 times. That's how big DNA actually is in our body. Now, remember, the real DNA would be millionths, hundreds of millions of sizes smaller than this, but it is actually that long. Okay, so let's go back and take a look at what we learned about our DNA. All right, so we know that there are actually in our body 3 billion pairs, and that this model would have to be 500 million times longer to make all 3 billion pairs. And so when we figured that out, it would be 37,000 miles long, which is about 1.5 times around the world. And your real DNA, this DNA is 8 centimeters wide, but the real DNA is about 2 nanometers or 2 millionths of a millimeter wide. So it's so small you couldn't even see it with your eye. So that means this DNA is 40 million times wider than real DNA. And that this long strand of DNA is coiled and folded into the center of every cell in our human body. So if I wanted to get somebody's DNA, one way I could get it would be from their blood. Another is in your hair or your saliva or your sweat. Those are all those places where I can pick up your DNA. Okay. Now, let's continue on and see if we can look at a model of half of the DNA, because remember, in the movie, they only got part of the DNA. They didn't get all of it, and so they had to fill in the gaps. And remember, they used a frog DNA to fill in the gaps, so they had to know what was missing. So let's take a look at this and see if we can figure out what's missing. Now, if we know that adenine only pairs with thymine, then if this side is A, what does this side have to be? Right, this has to be a T. And if this side is C, cytosine, then this side has to be G, guanine. So this has to be TG. If this is T, thymine, it only pairs with adenine, so this has to be an A. Guanine only pairs with cytosine, so this has to be a C. Cyanine goes with guanine, so this has to be a G. And guanine goes with cytosine, so this has to be a C. So if I didn't have this half, I could still figure it out. This would be T, G, A, C, G, C. And I can figure that out because I know the pairs and how they go together. So if I only got a partial or half of the DNA strand, I could fill in the rest of the DNA strand with the other half from using the frog's DNA, which is what the movie showed that it did. Now, we have only ever cloned one other thing in real life, and that was a sheep. And we did that several years ago. And through the cloning of the sheep, we found out that when you clone something, it's not as healthy as the original. And so that sheep only lived 3 years. And I know many people have talked about cloning people. And we found out that trying to clone people is very, very difficult. And they can often come out with lots of medical problems or malfunctioning of their bodies. And so even though we can and we know the science behind it, doesn't mean that it's the right thing to do. And one of the quotes in the movie Jurassic Park was from the dark-haired, curly-haired science mathematician, actually is what he was. He said, "We spent so much time learning how to do it that we didn't ask ourselves whether or not we should." And you can tell that in that movie, was it a good idea to recreate the dinosaur? Well, in the end, everything and everybody was destroyed. And so even though we could figure out how to do it and they made it happen, doesn't necessarily mean it's the right thing to do. So then why are we talk about DNA? Why do we talk about biotechnology? Well, because finding out DNA gives us a lot of other answers. Now, sometimes when somebody is born, they can be born with what we call a genetic defect, something wrong with their DNA. And that genetic defect can be passed down from parent to child. And so if there is a genetic condition in your family, a medical condition, and we want to find out if later in your life you're going to develop that same defect or illness, you can go to what we call a geneticist, a biotechnology geneticist, and they can test your DNA to see whether or not later in your life you would have that same genetic abnormality or the same illness. And they can begin treating you for it now. So very proactive. So that's one thing that we do. Another thing is looking at DNA strands to see if there's something we can do to tweak it to make it so that you wouldn't later develop some of these medical conditions. Another thing that we use DNA for is if somebody commits a crime, they can sometimes leave behind hair or saliva or sweat or a drop of blood. And from gathering those pieces of evidence, we can test the DNA, and that DNA will tell us exactly who that person is. Because no two people have the same DNA, that would mean that we could identify the individual who committed the crime. So DNA testing is something that is done often to eliminate suspects or to prove who might have committed the crime if a piece of DNA was left behind. So that's part of biotechnology, but by no means is it all of it. Okay, so let's talk a little bit more about our biotechnology and how we use it and what kind of jobs that you might get if you were interested in studying more about DNA. Okay, so let's go here. Here are some of our careers that you can do in biotechnology. You have a biochemist, a bioinformatics scientist, chemical engineer, a chemist, a cytogenic technologist, a genetic counselor, and a microbiologist. These are just some of the occupations. Now, if you are interested in this kind of thing, you could go on to Science Buddies right here. If you go on to Science Buddies, you can go into their careers. Now, let's say I wanted to know a little bit more about the genetic counselor. Say that's like a job I might be interested in. So you can see here that if I pull up genetic counselor, it tells you a little bit about it. It says many decisions regarding a person's health depend on knowing the patient's genetic risk of having a disease. Genetic counselors help assess those risks and explain to the patients and counsel individuals and families about their options. So this is one of the careers. Now, if you wanted to know more about that career, you could go in here to some what do they do, and here's some examples of some things they do. Key information. This tells you that how much money a genetic counselor could make. Here's $74,120 in salary a year. So that's a very high paying job. The average person is down here. This is minimum wage, and this is what a genetic counselor would make. It would tell you what subjects you might want to study in high school, what your degree needs. You need to have a master's degree. A lot of growth here. They're in demand. And you can see all of the more information about what education you would need, what would it look like to be on the job. You can watch a little video of that. Here's some different project ideas. If you want to do a science fair project dealing with biotechnology, this is some project ideas. And then there's lots more information on becoming a genetic counselor. So that's one of the many jobs that you could look at for biotechnology. So this is a great website if you want to learn more. Well, I hope you learned something today. I hope you learned a little bit more about DNA and biotechnology. I hope you enjoyed creating our model of DNA. And now, if you want to, you may eat your model. Just be careful and don't eat the toothpicks. All right, so I will see you next week. Next week, we're going to be talking about balanced and unbalanced forces. So the materials you're going to need are a glass or plastic cup, water, some quarters, and an index card. So that's what we would need for next week. Until then, keep wondering. Talk to you later.